Welcome to ASM Live from the 2014 American Society for Microbiology meeting in Boston, Massachusetts. I'm Stanley Malloy. I'm your host today. We're going to have a conversation about a lot of things microbiological, things that are important, things that are interesting, and things that are surprising. We encourage our studio audience to ask any questions that you have. Please come up to the microphone to ask questions. And if you're listening in online, you can send questions to hashtag ASM2014. Our guests today are Jessica Rivera Perez from the University of Puerto Rico in Rio Piedras, and Raul Cano from Cal Poly San Luis Obispo. Oops, sorry about that.
Happy and well.
And they have a collaboration between the two campuses to study fossilized feces and how fossilized feces can help us understand pre-Columbian cultures. Who would have guessed? Jessica, could you tell us a little bit about how you even began to think about doing this study?
Well, the idea actually came from Raul. He got in contact with the archeologists, Yvonne Narganes and Luis Chanlate from the Center of Investigations in Archaeology in our campus in the University of Puerto Rico. He found out that these archaeologists had access to these coprolites, these fossilized feces, and he basically stuck his neck out and the collaboration began. And then he began incorporating other scientists, bioinformatics, Dr. Steve Massey, the microbiologists, for example, Dr. Gary Toranzos, which is my mentor. And so we began trying to see if we could characterize the fecal microbiota of two pre-Columbian cultures that had inhabited the island of Vieques, which is in the eastern side of the main island, Puerto Rico, in the Caribbean. And so these cultures were a bit of a mystery up to a certain point. One of the cultures was well characterized, the Saladoid culture, but the second one, it was still unknown if they had the same origin of this Saladoid culture. And so since they weren't sure where they came from, they gave it the name Huecoid in honor of the place where the excavation site was located in the 70s. So what we did was basically characterize the fecal microbiota and see if we could determine major differences between these two cultures.
So Raul, what got you into it? What made you think about fossilized feces to begin with?
Well, besides being a fossil myself, I've been working on fossils since 1992, 93, and for a while, funding became kind of an issue, so fossils are not fundable. So unfortunately, we had to move on to bioremediation because that's what the action was at the time. But I retired in 2005, and I still have a passion for ancient DNA. So excuse me, this opportunity presented itself. Dr. Narganes was kind enough to let me share some of her jewels, and so that was for me. That was a perfect retirement present.
Jewels, present, feces. There's just something incongruent about this.
I am not particular.
So you have these two cultures. Were they physically separate enough so that you could distinguish which fossilized feces came from which particular one of these groups?
It is my understanding from looking at, discussing it with the archaeologists and also looking at the evidence that they co-inhabited the island of Vieques. Actually, their excavations were pretty close to each other, within walking distance. But they were still remain distinct and identifiable based primarily on archaeological remains.
They had different outhouses.
Yes. Well, I presume so, which is close to the garbage dump.
Dr. Naganes, she actually at one point commented that they were actually living a maximum of 20 meters apart from each other, but they were very separate. One culture had one part of the region to themselves, and then the other culture would have the other area of the region.
That's very interesting, just from a social perspective. But now, why don't you tell us what you can learn from looking at these feces? First of all, how do you analyze fossilized feces?
With patience. Well, first, what we did was we actually tried to extract the DNA various times. And at first, it was kind of uphill, but we actually got enough DNA for sequencing. And we did the bioinformatic analysis, and what we found was pretty amazing. I mean, the quantity of DNA that have been preserved in these species, I don't think we were even expecting such good quality DNA, and since these coprolites are not supposed to be well preserved in the tropics, then it was just an added surprise.
Yeah. But to add to this, our laboratory has had upwards of 20 years of experience working with ancient DNA. So we know what the caveats are. And we have tried over the years to improve on the quality and the handling of the DNA to avoid obvious problems that you may encounter with contamination, etc. And knowing that the DNA really is sparse at best, one has to do specific methods to be able to separate the DNA and ascertain that this DNA is actually from the origin, which we hope it's from. So, yeah, we've had DNA difficulties extracting because they're not much, but the quality is excellent, and it has a lot to do with technique and experience.
So presumably you have DNA from microbes that are in the feces, you have some DNA from the host, and potentially some DNA from other things that the host consumed in their diet.
Yeah, that's exactly what we're observing in the data.
And tell us, what did you learn the most from?
The most?
Well, so for example, one of the key things that you would like to know is differences in the diet of these two populations. Is that something you simply infer from looking at DNA signatures of plant products, or do the microbes also tell you something about that?
We kind of put both things together because previous studies have demonstrated that the fecal microbiota and the gastrointestinal microbiota are influenced by the diet of the host. But also, if you're detecting, for example, in our case, we detected maize in one of the cultures. Well, I mean, it's obvious how that maize got into that feces. So you put both data sets together and you start trying to develop a story.
But the approach has to be systematic. Okay, so first of all, the first question is, are there microbes in here, or is there any DNA in there? And once that question is answered, then you go to the next step. It says, you set up the null hypothesis. There is no difference between the DNA from all of these cultures. And then once you either prove or disprove that null hypothesis, which as in our case was, no, they are not the same, they are different, then you start asking for deeper and deeper questions and separate it microbiologically, bacteria, fungi, viruses, and functionally, and carbohydrate digestion enzymes, lipid processing enzymes, things like that. So it has to be a very systematic approach, because if you just try to encompass too much, you'll have a descriptive study, but not substance.
Another thing is that one of the first things that we did when we got the data was to see if the microbiota that we were detecting is normal gut microbiota, because since these species were exposed to the environment for so many years, for about 1,000 or 1,600 years, we didn't want to be deriving conclusions based on soil bacteria. And once we saw that the microbiota that we were detecting is representative of gut microbiota, then we were more confident with what we were obtaining.
So before we go on, I want to remind our studio audience, please join in on the conversation at the microphone at the side of the room. And anyone listening online, please send questions at hashtag ASM2014 via Twitter. So what are the main findings, the differences between the two groups that were really important?
Okay, the main findings are the microbial groups that are representative of the human gut, the gut microbiota is there. That's important. The next is that one can actually discriminate the cultures based upon microbial community analysis, the standard analysis, the alpha diversity, beta diversity, and statistically significant difference in the distribution of microbes in the two cultures. Enzymatically, I'm not sure yet. I know that there are some differences in which the abundance of carbohydrate active enzymes, but this is very preliminary. But this, from the point of view of microbiological, yeah, you have the baseline is, yes, it is likely to be a microbiological community of the feces. And there are differences in relative abundances and representative groups between the two cultures. So it suggests, statistically at least, significantly different between the two cultures.
And that potentially suggests a very substantial difference in diet.
It certainly does. It certainly does. And I think that the more we know about the metabolism and we delve more deeply into the microbial community structure, we'll be able to say something like that.
And one important aspect is that many archaeological studies have also been done on these two cultures. And they have also been detecting differences in diet as well. And so what we're seeing is not just basically an abomination. This is backed up by other studies, morphological studies, and also social studies that have been done previously.
So you mentioned earlier corn as being associated with one of the groups versus the others. What's the significance of that?
I'm not sure I understand.
You said you found evidence of corn in the feces.
Oh, right? Yes. Well, corn is basically has been proposed as one of the most important parts of the diets of many of the pre-Columbian cultures that have inhabited the Caribbean in one point or another. And so in the archaeological circle, as far as I'm aware of, many studies have been done in trying to understand which cultures consumed this corn, which cultures brought the corn, etc. And so, for example, previous studies have found corn remains in, for example, artifacts that they use to grind their food. And so it was very interesting for us to find corn DNA in there because this is basically supporting previous findings with molecular data.
And so that corn doesn't really distinguish the two groups. Both groups consume corn.
We believe that the fact that, we detected corn in one of the cultures, but this is preliminary data. And so we believe that the fact that we did not detect corn in the other culture doesn't mean that they didn't consume it. Maybe they didn't consume it as regularly as the one that we did detect corn.
Well, not to be real contradictory, but we really did find corn in the other culture as well. Just the relative abundance of that gene, that information was greater in one culture than in the other. But it may have also, we have not analyzed that in depth, because it may be due to relative abundances because of diet, or it could be just because in one, that gene was preserved better than in the other condition. So it is difficult really to make I wouldn't go out on the limb, say, you know, they eat corn and they don't, because I'm sure they all did.
So the reason I asked is it would be interesting to say, can you correlate where the group came from and the type of diets that were typical for the two groups migrating to the same spot, going backwards from the feces to predict what happened prior to that time?
It appears that, you know, the diet issue is a little more global than that. You know, I think they had some. I mean, again, I wasn't there, damn near, but wasn't there. But it was. I'm thinking that they had a set of staple food that they would use, and some of them they have preferential, like Cubans prefer rice over potatoes, yeah, and I'm sure some prefer yam over cassava. Or corn, and for that's why the corn wasn't as abundant in one culture as in the other. So the issue is that I think you can, looking at overall, you know, presence and absence or relative abundance of these groups, you might be able to tell diet and dietary preferences. Definitely, there are definite preferences in protein source.
But one thing that we were talking about, and we would like to, you know, reach out to other archaeologists in the field and see if they have the samples. We would like to see if the particular species of corn, for example, that we were detecting may be somehow correlated to particular species that grow in certain regions of, for example, South America. Mm-hmm. But that's future studies. We would like to do that, but we don't know if we would have the opportunity to do so.
This seems like a great approach that you've looked in Puerto Rico, a very important place for migration, but you could apply this approach very broadly around the whole world to understand human migrations, I would think. I
think so, and I, you know, the Caribbean is a real, very close to my heart. You know, I was born and raised there, what happened in Puerto Rico and what we have seen in Vieques is happened all over the Caribbean. And because the general consensus of the scientific community is that coprolites cannot be found in the tropics, these studies have not been pursued. Hopefully now that, you know, Dr. Narganes and Chanlate have been able to very successfully demonstrate that you can get coprolites, then we can take the next step further and bring the entire community from the Caribbean to search for and start analyzing these coprolites. And that's really my goal, you know, my long-term goal. I'd like to be able to achieve that goal, at least, you know, have colleagues looking into that possibility.
Very interesting. We have a question from our online audience.
Actually, we have a couple of questions from the chat room. First one comes from Hannah, and she asks, so did you just throw out any bacterial sequences that were not known as modern human gut bacteria? Doesn't this assume the human gut microbiome has not changed over this time period?
Literally the only sequences that were thrown out were the bad quality sequences. We did not introduce any bias in terms of, oh, well, these are gut associated, so let's just use these sequences, and these are not, so let's just forget about these. That wasn't done.
But let me add to that. Did you find some sequences that you couldn't place when you look at the databases of the current gut microbiome?
There were plenty. You know, what can you do with something like that? Eventually, you can, you know, isolate those sequences and study them further. But, you know, there is a core microbiome in all human guts. And that core microbiome may change in relative abundances, but it doesn't change in composition. So, you know, you do have certain fecal representatives like Bacteroidetes, the Clostridiales, and all those organisms that are present, but not necessarily in the same relative abundance or the same species representation.
Another question.
Our second question comes from our good friend Gary Toronzos. And he
An
wants
insider
to know.
question.
We don't answer.
Yes, I believe he's also an author on this study.
Yeah.
What about the parasites that were also found in the coprolites? Not only this study, but in previous ones as well.
Well, one of our collaborators, Dr. Erilyn Garcia, she actually did a parasitological study, and she actually found, and this was completely independent from our study and previous to our study, and even at a parasitological level, she found great differences between these two cultures. She found that, for example, the saladoid culture had a greater diversity of parasites, whereas the wecoid culture had a smaller diversity, but both had parasites. But a major difference was observed as well.
Was the smaller diversity accompanied with a higher burden from as much as you can tell from any kind of quantitative guesses?
It depends on the parasite.
You know, from her work, it looks like Enterobius was very prevalent in one culture versus in the other, and almost non-existent. But a larger parasite diversity was found in another culture. And we can actually, looking at data that has not been presented yet, it appears that her findings are being reflected in our DNA analysis, which is nice to say.
That's interesting. Now, sometimes, if I understand, not being a parasitologist, if I understand, certain parasites can be associated with certain food groups more often. Can you infer that from these studies, and to tie into your work?
Go ahead.
Well, for example, with our data, we found a particular parasite that infects fish. And so when we look at the suggestions that archaeologists have proposed in terms of the diet of that culture, it correlates perfectly. For example, we also found a parasite that infects crustaceans. Again, archaeologists found a large number of crustacean shells in these excavation sites, and so it correlates as well that we find a parasite that infects these organisms.
It really is a nice correlate to the other things you found. Have people begun looking elsewhere at DNA and coprolites to try to build stories along these lines?
There has been a few studies with coprolites, human coprolites. Some of them have focused on human gene and trying to use it as a way of, for example, the clothes man, trying to trace his or her origins, and that has become quite a hot potato because it's really difficult to separate modern from extant. But I assume that they did their work as well as we did ours, and so it's terrific. And there's also other groups that are actually looking at much older coprolites of 15,000 years, and they're looking at the microbiological components. So it looks like there is becoming an interesting and the new thing to do in archaeology, or in paleomicrobiology. I presume that that's a better term. So it is exciting that we are there at the beginning. And at least we can hopefully establish some standards of best practice or contribute to the development.
So I think what you mentioned ties into an earlier question of basically, has our microbiome evolved over the last, in your case, about 1,200 years? And you could compare it with 15,000 years and potentially go quite a ways to see if, in fact, what we see in our microbiome now is pretty similar to what was seen in those days.
One of the things that we are doing now is looking at the published literature describing microbial communities, as well as enzymatic composition of the gut biota, and looking and comparing it to what we're seeing, we have to deal with the fact that we're dealing with ancient DNA, so there's information loss along the line. But so one has to, that's why I mentioned using the core microbiome or the core data to be able to make comparisons. So yes, I think this is one of our goals, mid-range goals. Right now, we just get this paper published.
We have another question from the studio audience.
Hey, how you doing? It's very interesting work. I was wondering if you have this European population, you have the population that you've been looking at. One might imagine, even though that microbiomes are very stable, they could be these two populations that were separated since Africa are now coming together at some point. Is there a possibility to see how these biomes will change over that type of intermixing of peoples?
The answer is probably yes. I'm not going to say definitely yes, but you know, the thing is accessibility to samples and have a sufficiently large sample size that one can actually make some statistical or at least some reasonably based comments. I was just listening yesterday to a poster in which you need require, like, you know, for a data set like ours, we would require like 15,000 or 14,000 samples. That's never going to happen. Yeah. So, but yes, I think it's a possibility. It's a lofty goal. Yeah.
But an interesting aspect of that question is these two groups that were separated by 20 meters. It's phenomenal. You can have two distinct social structures for a long time that are that close together. But presumably, as time went on, either they intermingled or one disappeared. Do we have an idea about that?
Well, Dr. Narganes has commented that, in fact, what they know about the Wekweid culture is still so little, and they still do not know if this was a very, I don't know if the term would be a resilient culture that didn't really, for example, they wouldn't marry off to people from other cultures, in contrast to the Saladoid culture, which they know, for example, would learn different techniques from other cultures and would probably have a much better co-fraternization. They still do not know what was the relationship between these cultures. A lot of studies are still needed. One hypothesis is that probably during the time that they cohabited in this area, obviously they learned from each other, and you can see that from the artifacts that that these archaeologists extracted from the sites.
We have another question from the online audience.
Yes, Gary wants to ask another question. Would you consider extant feces to be more homogeneous than ancient ones? Because McDonald's is everywhere in the modern age.
I would say, I guess it would depend who deposits it, whether he goes to McDonald's or not. But I would disagree, I think, of the homogeneity, because, you know, even when you go to New York City or Boston, there are neighborhoods, and they're, you know, this neighborhood, they're all, you know, even though they could be interlinked, they actually have different food habits, and that would definitely impact the microbial flora. Sure, I'm talking 1955, microbiota, the gut microbiota of the individuals. So I think that, no, there might be homogeneity among certain segments of the population, but there's also going to be cultural differences in isolated communities.
So it really gets to be this interesting question of that I think is a question for thinking about the microbiome nowadays that reflects how does it change. And since we know the microbiome can influence health, mental status, everything about us, to think if it was a mingling of the microbiomes of these two communities, what that would have done to the culture and society in these groups. Mm-hmm.
Well, in fact, I think there's a paper by Dr. Dominguez, Maria Gloria Dominguez, today. It's talking about the gut microbiome across cultures. I mean, I haven't seen the poster, but I think I looked at her results, the data, and there seems to be some really important differences in the core microbiome and the gut microbiome across cultures. So it is inhomogeneous in that respect. I'm looking forward to seeing what she had to say.
So we are about ready to tie things up, but I want to thank our guests again, Jessica Rivera Perez from the University of Puerto Rico in Rio Piedras, and Raul Cano from California State Polytechnic University in San Luis Obispo.
Amen.
Our Cal Poly San Luis Obispo.
Good.
And they told us a very interesting story. This is one of those examples of a concept that we wouldn't have thought about so long ago because we wouldn't have had the tools to really begin to dissect this. And it kind of shows how microbiology is having an impact on so many areas of science and thinking about life. It's a very exciting time we're in, as long as we can keep getting funding to do it. So we want to thank all of you in the studio audience and all of our online listeners. Once again, please join us for the next version of ASM Live. Before I sign off, let me add one other thing I almost forgot. Yesterday was the very first version, sponsored by ASM, of ASM in Vivo, a Spanish broadcast talk show of interesting things microbiological. So if you're listening from the Spanish-speaking world, look up the archives of ASM in Vivo and look for future editions. Thank you very much.
Explore the fundamental role of microbes in the natural history of our planet with Microbes and Evolution, the world that Darwin never saw. Available at estore.asm.org.